Hi, my name is Alexis Ellis. I go to American University, and this summer I was interning at the Frank and Stroke Lab working on building an automated tissue processor. Now, my project is centered around propydium iodine staining, or PI staining. PI is a dye that plant scientists use in order to visually see a cell wall more clearly. This process is usually done manually, and it's very time-consuming. Now, the Frank Lab utilizes this method quite frequently. One member of the lab even expressed to me that last summer they did over 150 PI stains. To address this issue, my project combines engineering principles as well as biological processes. Now, there are automated tissue processors out there. However, they are not cost effective and can range anywhere from $10,000 to $15,000. This makes this matter urgent because we need to find affordable alternatives that can be replicated in other labs as well. My project seeks to make plant science research more cost-effective and accessible so that way we can increase workflow. One of the main focuses of my project was making sure that it was affordable for other labs to replicate. In our hardware section, we have 4 key components: our relay, our Arduino, our peristaltic pumps, and our solenoid ball valves. No one part ranges more than $30, so that way it can be affordable for other labs. Below that, we have a chart. This chart displays the volume compared to time for our peristaltic pumps to pump milliliters of water. As you can see, for about 90 seconds, it takes 100 milliliters to pump. Moving on to software. To conduct my software, I utilized a graphical user interface. Now, this interface was comprised mainly of Python coding. Whenever a scientist would be ready to conduct a PI staining, you simply press play and then a pop-up window displays. In this pop-up window, it looks similar to this one. It asks the amount of steps that the scientist would like to conduct. Here we have 3, and then 3 sub-windows would pop up asking for the percentage of ethanol you want to display. The prototype can display any number of ethanol from 15% to 100%, and even DI water. We implemented this graphical user interface to make it more user-friendly, and so that way scientists don't have to interact with code every single time they want to conduct a PI staining. Additionally, our Python code communicates to our Arduino code. Arduino utilizes a language called C++. The user implements data into our graphical user interface, and then the Arduino then receives this data and then sends the desired signal to the pump or valve you want to control. Moving on, we conducted an actual PI staining with our prototype just to show that even though we were doing it in a lab, it can be implemented and actually work on different types of stem and leaf cells. So here we have a fig and we have a tomato leaf. We were able to yield successful results with our prototype, going through the whole PI staining process and even producing successful results. In our fig and tomato leaf, you can see that we can visually see the vasculature of the cell wall and as well as the mesophyll, which shows that our prototype did have successful results and can be implemented in the lab in the future. In conclusion, I spent this summer building an affordable and easy to use graphical user interface as well as prototypes, so that way we can minimize the amount of time and the amount of work that scientists go through for a typical PI stain. This whole automated tissue processor is also affordable, costing no more than $250. This will also be implemented in the Frank Lab later this fall, 2023, for further testing and redesign. And upcoming actions that this prototype can take are making sure that our pumps are more robust. Although it is linear, the pumps can work faster to receive the desired amount of milliliters that we want to produce, as well as adding a mixing feature to our tissue container to make sure our ethanol and our water never sit for too long together. and we can always further improve upon the graphical user interface to make it more user-friendly towards the Frank Lab members. Additionally, I would like to thank the Frank and Stroke Lab for allowing me to intern with them this summer and create such a meaningful project. I would also like to thank Elise Bovart and Michelle Henney for guiding me and introducing me to a typical PI stain, and thank you.